picture is on the wall.
So I will introduce, I'll set up Amy, who's going to talk to you about the science and pose some of the questions. So, you know, I could put on my sarcastic hat and say, unfortunately, this won't work for plants or food because it's all DNA based and neither of those have food, right? Kind of honor Whole Foods opening up down the street. So this is a really cool application that you know, we've been thinking about for a while, and actually kind of part of the idea, the enthusiasm for me came with it on a trip to Sri Lanka with Chowani a few years ago, looking at people talking about their data and how they're using it and realizing that there's so many generations behind us. And my lab, I'm going to go through just a few kind of general ideas of what kind of the buzzwords that we're thinking about, really kind of field deployable genomics for plant health, for pathogen traits, and really it can be anything with DNA. So if you have DNA in your project, this is accessible. So I want to just give a few slides about what my lab does and how we kind of got involved or interested in this, and that'll kind of get you to wrap your brain about how I look at this, how I look at this problem. So my lab's very interested in these types of analyses, looking at the plant pathogen interface, and we collect a lot of data on when certain genes are expressed. And I promise there's a point to all of this. So we can analyze a lot of genomic or RNA-seq data, and we know every single gene that is expressed at every single step of the infection, including the pre-symptomatic stages of the interaction. And that's where I think the real power and opportunity for this project comes. And so as we were collecting all of this data, and for those of you that have labs, or for those of you that are students in labs, which I think covers everybody, you realize that when you generate large pieces of data like this, you can either do two things. One, you can talk about all the genes at kind of a superficial level, or you can work on one or two genes at a very deep level. For those of you that work on one or two genes at a very deep level, you've got oftentimes between 29 and 50,000 other genes that are just put into a hard drive, filed away somewhere, and you never look at it. And on a trip to Sri Lanka, I was thinking about what is the value of this? We know everything about this plant pathogen interaction. So this is downy mildew of cucurbits. So from an experimental standpoint, this is a very complicated place to start. It's an obligate pathogen that can't be transformed in a host that's really kind of an orphan crop. It's important around the world. India, China, Southeast Asia, you can find dozens, if not 50, close to 100 different varieties of cucumbers or cucurbits, melons, cantaloupe, blah, 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 that are eaten all over the world. And we can do these types of analyses, and because it is an obligate system, every time we take a sample, we're collecting the pathogen and the host. So we know the expression of every gene of the interaction at any snapshot in time. And then we can start looking at things about, okay, when does symptomology occur? When do we start to see symptoms? And really that gets into the question of when can we detect the pathogen? And in the lab, you've all done real-time PCR, I'm sure. These are not field deployable methods. They're very robust, they're very specific, but they take 2 to 3 hours to do. They're expensive. The current state of the art in plant pathogen diagnostics is there's a few immuno-based kits, antibody-based kits. You grind the sample up, you process them in the field, you do an ELISA assay, and these are between $3 and $5 to $8 per reaction, per plant, per sample. And there's a lot of cross-reactivity. Antibodies can cross-react with each other. So this gets back to the resources that my lab has developed, and many, many, many other labs around the world have developed, is we've got tons of genetic information, tons of DNA-based information available. And so what we started asking was, how can we deploy this? How can we do this in, a robust fashion that it doesn't cost you anything to develop this assay, because if you've got a computer, you can access PubMed databases, gene bank databases. You can create markers, blueprints, fingerprints to detect whatever you're interested in. It can be a pathogen, it could be a trait in a plant, a gene that you know is involved in a certain process in the plant. It can be a transgene, so if you're a plant breeder and geneticist, plant breeder, for example, and you're out in the field and you're making crosses and you're looking for a specific trait, and you know there's a gene that segregates with that trait, you know the sequence of that gene. You can find the sequence of that gene, you can track it. So what we really wanted to do was take advantage of what's out there, so $3 to $5 per assay, There's a little cross-reactivity. They take anywhere from 1 to 3 to 4 hours to develop, sometimes longer if you send it to a diagnostic clinic. And oftentimes decisions are made in a matter of seconds. A decision is needed in a matter of minutes. So how do you get there? And cost is one of the big things. So $3 to $5 per plant. If you've got 15,000 acres, that can get kind of expensive. The assay that Amy's going to tell you about that she has developed in the lab, not counting her time, but okay, if we add in her time, it adds an extra nickel. So that's the reality. But one assay, $3. Imagine if you had an assay that could tell you everything you needed to know about the trait that you're interested in or the pathogen. And what if I told you you can do 40 of those assays for 1 penny? And so this is where Amy will come in, and it is building on a case study that kind of was a concept really. And so it's utilizing genetics, nanotechnology, a little bit of, I threw the biophysics in there to capture kind of the linkages to many of these applications and devices. The processing, the matrix. So it covers all of biology. Right now we're doing plant leaf samples, but milk, for example, is a big problem. Neogen is very important. When I was in Sri Lanka, we visited a natural food processing lab, Institute of ITI, right? ITA. Institute of Technological something or other. And it's a natural food processing lab. So there's a lot of different matrices. It's not a nice pretty leaf that has a single pathogen on it. It's food, it's lotions, it's soaps, it's all of this stuff. So there's some accessibility things that I think as biologists and engineers we need to think about. How do you know, it's easy. We can do a real-time PCR on a leaf. How do you do it on Suave shampoo or lotion or something? Communication, this is very, I think is one of the underlying principles of this technology, of this network, this social network. So I'll let Amy start here with kind of talking about how she adopted a case study in the lab and developed a really cool nano biosensor assay for detecting DNA.
Thank you. So, pathosystem, being a plant pathologist, that I started with was cucurbituril downy mildew, and this kind of gives you an overview of the epidemiology of this. And the reason we focused on this, besides Brad's lab working on this prior to me starting here, was also it's very important to Michigan and the industry of Michigan agriculture. As you can see in this map here, kind of the spread of downy mildew, and how this starts down here in the south during the winter, where they propagate cucumber, and then it'll spread throughout the summer, probably through moving transplants, shipping up to the northern region where we'll see it in May through September. So this is a very broad spread and impacts the whole nation and into other areas where they produce cucumber. So this is the pathosystem that we started with. However, there are other pathosystems and pathogens that we're interested in spreading this, or using this technology for. One was being frog-eye leaf spot of soybean, which has fungicide resistance to it and is developing and spreading, so this is more of an economically important pathogen to field crops. Also soybean sudden death syndrome, which is a pathogen I'm going to be working on for another portion of my dissertation. As you can see up here, it started in Arkansas, and over the last 30 years it has spread throughout the U.S. and major soybean producing areas. Also Goss's wilt, corn, and you can see that that's a major emerging disease as well, and that's a bacterial disease, and that is affecting all major producing areas of corn. And part of the problem of this is that we have global movement. Import-export trades, we're a global society now. This is a map of the air traffic and flight patterns. But so just imagine, one of my friends actually this week, I used to work for a potato company in the industry. They flew their transplants from Idaho to Michigan, 2 hours. What if those had a virus? That's really moving that from the western U.S. to Michigan. But what if you don't know? What if it's latent? You have no idea that that's going on, an asymptomatic infection. Another question of this, or problem, is regulations, trying to quarantine these pathogens from moving, and they can spread within seeds that that we need for planting. Weather is changing currently, climate change, so that's going to affect our diseases and how they spread. And then people, our interaction, that's a high interaction. We're changing the agriculture that we have today by what we're selecting for. So to understand these evolving processes that are going on globally, we're looking at using a DNA nanosensor. This is understand, it works with the DNA of whatever you're interested in, plant, fungal, or bacteria, virus. And generally, within the time frame of having a question, I drew clover leaf here because it's easy to draw with some latent, some chlorotic areas to represent our pathogen. Being a plant pathologist, I work with pathogens, but you can also be looking at beneficial microbes and just anything. But generally, the question is, you can go from question to detection. Right now in the lab, with what I'm working with, of extracting DNA, running this assay, you're looking at about 2 hours total. Now that's doing extracted DNA. This is proof of concept stage. So there's a lot of optimization to be had yet. What I would expect this to encompass in the field would be 30 minutes or less. Once we get that matrixing down and working with leaf samples and tissue samples to be able to grind that sample with your plant and microbe, you would then have your DNA in a solution, a lysis buffer. Then you have your probe. So think of how PCR works. You have a primer that is very specific to your target. And when you heat up or denature the DNA, it will then anneal and bind to your target. If it's a non-target, let's say like a plant tissue, it won't bind at that time. And then you add your nanoparticles to that solution, and that will bind to the probe or your primer that is in solution, because nanoparticles, these being gold, have a very high affinity for single-stranded DNA. So the single-stranded DNA will be absorbed to the nanoparticle, and then you add salt, which causes the nanoparticles to aggregate and turn blue if the primer is bound to them, or stay red if they're not bound. So to kind of get into that mechanism, because that's kind of a unique mechanism that's occurring, right now in the lab, how this functions is you have your target DNA and probe and non-target DNA and probe, and you add that to, let's say, a PCR tube. Right now I'm working with a volume of about 20 microliters. You denature that and anneal that, and then you add your gold nanoparticles, which are actually red, and then you incubate that and add sodium chloride, which is your aggregation. That's kind of what defines that detection. And then if the target is bound to, or the probe is bound to the target, it will stay pinkish red, which is the color of the nanoparticles. If the probe is unbound and in solution, it'll turn purple.
So at this point, just to kind of start a conversation, the processing is the longest part of the assay. From the time you initiate the colorimetric reaction, naked eye, it's less than 5 minutes for you?
It's about 5 to 10 minutes, yeah.
Okay. So spectrally, you can probably begin to see that separation in color change really quickly, I would imagine.
Yeah, so this part, as Brad said himself, is very quick. It's the DNA extraction part right now that takes the longest. So to kind of give you a visual of what we're looking at here, this is our downy mildew or cucumber pathogen that I was originally working with. Thank you, Brad. So this is with 2.5 nanograms, which is kind of after a higher concentration range for DNA. So this is our control. I just add water rather than DNA in here. A non-target being some corn DNA that I had laying around the lab for working on another pathogen system. And then our P. cubensis, our downy mildew cucumber, our DNA, that's our target. And what you see is that that's a little pinker, and then these are a little purpler. But however, to quantify this more, and to give you a better grasp, as scientists, we all like numbers. We have the spectrum. So this is the UV spectrum. And this is absorbance, and then it goes from 400 to 700 nanometers on the bottom. And gold nanoparticles have a peak around 525 nanometers. And that's kind of where we use to assess concentration. And that's where that middle peak, or that high peak, is. And then as you look to the right, you'll see that the gray line, which is our P. cubensis, our target DNA, drops lower, which means that as they're turning that kind of purple or blue, that's where that absorbance value will fall around 620. So there's going to be a higher value at 620 for our non-target, our control samples. And then if you take that ratio of 620 over 520, then you can kind of quantify that as the shift. And so our control and corn being non-target is higher, much higher than the P. cubensis. So kind of illustrating that difference. You okay?
Well, never mind, I'll come back later.
Okay. You look like you had a question.
No, not a question.
Cameras in. Okay, so then utility of this. Where can we apply this? Where can we go with this technology? And the big thing is to integrate this into our research. As I said earlier, I'm starting to optimize this to my dissertation research. Our main question was soybean sudden death syndrome and optimizing this technology to that. And it's broad accessibility. This is easy to work with. You just need nanoparticles, salt, DNA, a boiling pot of water to denature that and let it cool. That's it. Connectivity. So with the PhotosynQ device, we can connect this and relay this information for social engagement for everyone to understand, oh, I have this pathogen here, I have this microbe here. And then understand how diseases spread and move. Owner responsibility. So to understand, like if I'm a grower, and I want to report this so that other growers in the area know how to manage their disease. As Brad mentioned, this is very affordable. And at 1/40 penny of the cost, these nanoparticles, even though they're gold, we can synthesize them in the lab. I can create them myself. And it's very adaptable. And as I just illustrated earlier, I'm talking about, I'm adapting this to my own research right now. As Brad mentioned, if you want a marker to look at for breeding, you can look at plant DNA, pathogen DNA, other microbes. All you need is DNA. Any questions?
Done?
Yes.
Could you go back to that slide I was?
Yes.
Last time we talked, we got really excited. We said, okay, I want to see this spectrum because maybe we want to figure out how hard it will be to measure it with this device.
Yeah.
But we already have those wavelengths. I would do it at 550, about 600, 610 nanometers, and 650. Your sensitivity would be higher than doing it with 3 wavelengths and 2.
Yeah. Well, this is what's commonly, for publishing, what's commonly used
Yeah,
for this
but
type of asset.
those wavelengths are already in here, so if you have a sample and you bring it down,
Absolutely. I mean, that's the missing gap here is the connectivity. So in the field, we've talked about, okay, how do you package this? Because, it seems like there's multiple moving parts. There's really not. You could probably do a chemical lysis step, take a small leaf punch, just go out and grab a leaf, put it into chemical lysis, suck it up, add it into a tube that already contains your gold nanoparticles with your primer, let them go for a minute, add salt, boom, suck them in, capillary, you're done.
I think we need to do all that, but
You gotta do some of it.
spectroscopically, this is really easy.
Yeah.
You've got everything you need already there.
So the device is mainly just to look at the color differences.
Sure, that's all it is. And the device, like Dave was saying when I talked to him earlier, the device can probably see it faster than the naked eye,
Yeah.
right? She's sitting around going 5 minutes. It's like the pregnancy test, really. I mean, that's what it is, right?
Except for less scary.
Yeah. And it's 2,000 times cheaper.
Depends on the crop.
It's 2,000 times cheaper than a pregnancy test, right?
Right.
People will pay $8 for a pregnancy test. You can do, for $8, you can do 320, is that right? Or a lot?
A lot.
These aren't the kind of nanoparticles that fluoresce. These aren't the quantum dot kind of things. These are non-fluorescent.
These are functionalized nanoparticles that are kind of proprietary, but.
Yep.
You make them anyway. So
Well,
you make
we're
them.
working with the collaborator that
Oh,
owns the
okay.
patent, so
Oh.
MSU has the patent.
So why nanoparticles instead of coupling like a fluorophore and a quencher just to your DNA segment?
Because nanoparticles have a very high surface area. There are, so that way they're more sensitive.
Okay, so you can get a lower detection limit.
Yes.
Okay.
Yeah. And the really cool thing about this, and Amy, we were concerned about this when we first started doing it, was with genomic DNA, she seems to be getting even lower limits of detection. Is that right?
Yeah, PCR and part of this is, if you go here, so part of with genomic that is more sensitive, I believe, than PCR. So your PCR fragments I'm working with for this gene are 422 base pairs. And so there's one target in there. But with genomic, they're spread out. You have this is my mitochondrial DNA that I'm working with, and there might be multiple copies of that within the genome, within the cell, and also that there's they're spread out. It's not like I have short 422 base pairs. So when these nanoparticles interact, and I mean this is more of a detail than what I explained in the presentation, but what we believe, and as looking at these through TEM, is that these nanoparticles are actually interacting with the single strand of DNA that form when you get that primer binding. So you get like a triplex form, and what they call these are DNA bubbles, because you have that bubble, because you have these long strands of genomic DNA. And so these nanoparticles are then sitting kind of on these bubbles, and that stabilizes them from aggregating and protects them from the salt, because they're separated. And so you're going to get that with genomic DNA. With PCR DNA, you're just going to get these short fragments, and they don't get that stability.
So bigger is better.
Yes, it's we call it.
We always eat.
Yes.
Are you worried about like spliciforms and whatnot? Like how sensitive are these particles? Like if there's like a nucleotide difference, are they still going to bind to that?
There is literature showing that they can detect single polymorphisms. Yes.
Damn.
Sounds like it would depend on your probe that you add.
Yeah, so all of this, and Brian's correct, it's very dependent on the specificity of your primers. And so they have literature showing that this is how they detect cancer actually, is with this gold nanoparticle assays and the oligonucleotides. And so maybe you might be able to get that sensitivity, like for that fungicide resistance on frog at least spot soybean could be something we apply that to look for isolates that have that.
So, yeah, so, you know, with genomics, the point of what I was showing is we're generating so much data and so much, you know, different, I mean, we're drilling really deep. So we can mine that data to find those short little 25 nucleotide through K-mer analysis to identify the little fingerprints that we need. And you can multiplex with this.
I'm wondering if the dose response is critical. In other words, you've got this step where you're extracting your DNA and processing it, and then you're going to mix it all up, and then I guess somewhere you have to normalize the amount of DNA you add. Is that right?
Yeah, that's a bench experiment, right?
But I mean, out in the field, is that going to be?
That's something we'll be looking into over the next couple of weeks of working with, starting with leaf samples and food samples to see if there's an impact on the response if you have a higher amount of DNA present.
So from a kit standpoint, you might just include a little lancet in the kit, say, take this sample, take no more, you don't need more because we know that more is not better.
You get differences in extraction efficiency.
Exactly, so then that comes into the kit. Maybe a chemical extraction would be.
Yeah, and they have that with the ELISA where you're not supposed to add more than X amount of grams or volume per lysis buffer to keep the ratios correct.
So as long as Bubba can read, this is kind of bubble proof.
I was wondering if you could put in a reference nanoparticle.
Yeah, and that's something that Brad and I have been talking about of
Pregnancy test, first strip means you got the stick, second strip means.
like a plant, let's say we're looking at this for a plant pathogen, so we have a plant gene that's in there, it'd be like, yes, this, is working. We detected a reference gene within plants, and then you have another reaction saying, yep, look, it is working for the pathogen. One thing, like in the pregnancy test, these are called lateral flow strip tests, and this could probably be incorporated into a format like that as well if we didn't want to use the the spectrum.
So maybe some adaptability with the Colorado State, lateral flow type reaction. Embedding the nanoparticles in a lateral flow, and then as they hit these nanoparticles, they separate out into these different wells, and there's your multiplex. So it's pregnant, female, will be 5'2", all of these traits that we can.
It's gonna have blue eyes.
Blue eyes. Yeah.
What he's referring to, by the way, is this, we were hoping to have another visitor here, but couldn't make it. A guy named Chuck Henry who does paper microfluidics.
Okay.
So you can build, and very inexpensive, but very good things you can do is get the separation. Anyhow, but another question for you about, can you get different colors?
With gold?
Nanoparticle
Yes,
colors?
yeah, there's like
That's
cadmium,
what I'm
zinc,
talking about.
copper, and they're all different colors.
Yeah, everyone has a different wavelength that they peak at, so you can just add a tube and have 10 different nanoparticles.
How do you ensure the nanoparticles are associated with different things?
That would be
So they have different affinities, and I don't know if we would actually mix them because we want an output.
Or just take your
of-
sample and repeat pipette
Yeah.
it or something.
Because if you want, if you're looking at silver, let's say, that has a yellow color shift from yellow to, I think, kind of reddish when they aggregate. And so you wouldn't want to mix that with gold because then you couldn't tell a red to blue
But
color
then if
shift.
you got a lateral flow system, you pop in your sample at the top, sample flows through, it binds the primers, and then it hits the wells that contain each one of the different colored nanoparticles, and then you just
So you
scan it.
can
in the
maybe
system.
get a rough idea of how many copies of DNA you're putting around there then?
Yeah.
So I have a question about sensitivity. So you're going to have a slight shift in blue and red, all right?
Yeah.
And you're doing this in like a 20 microliter reaction, like a PCR2, and you're looking for just like a slight shift. And so you might not have any sensitivity issues at all, but I'm wondering if it would be easier to have like a gold-coated little bit on the tip of a needle or something that you dip in, and then you're sensing the sensitivity on the tip of whatever, rather than it in a complete mixture. You're isolating the sensitivity. So rather than all these gold particles floating around in solution, they're all isolated and a nice, fluorescence is higher when it's aggregated.
Yeah. Yeah, that'd just be figuring out different concentrations of the reaction. So yes, it could be possible. And that's how with the lateral flow strips, they are concentrated in one area and then they disperse. So yeah, because with these originally I was working with a different set of nanoparticles, and they're part of the reason why I switched to these ones, which enabled this reaction honestly. But it was hard to see; they were too dilute, and so these were actually a little bit more concentrated, and they were easier to visualize.
So it's kind of like speed dating. So we're looking for people to partner with. If you've got DNA in your project, we're interested. We're going to start working with Carolyn Malmstrom and Dave Douches on viruses. Because viruses are completely different beasts, both of their interest in RNA viruses. It's just a different probe design. And if we can get that to work, which I'm pretty confident we can, that blows open plant pathology, because the primary diseases affecting plants are viruses. And then it becomes amazing, I think, because a lot of the kits that are out there now, I think Adgia, for example, has a kit, but it's got an extra enzyme step to convert the RNA into DNA. So there's always an amplification step. This is non-amplified. So when you start talking to people about DNA diagnostics, there's always an amplification step, whether it's a lamp assay or something. And that requires you, those are field deployable too, as long as you've got a cigarette lighter, you can plug it in and have a little box there and heat it up. But this is people in tribes and Mayan villages, civilizations could do this.
Is it quantitative at all? Like, could you get the ratio of, say, 2 different microbes in a system?
We've seen that.
With this current optimization design, right now it's for a single microbe. I think you could. I was just looking at a paper today where they had 2 different probes in the solution, and if it attacks, I'm sure you could get like a different ratio here of like, okay, if they're all bound, you'll be way down here, here's 1, here's 2. It might not be which one per se, unless you're looking at, you could do different nanoparticles.
Because you could get an idea of like how much of a particular microbe is in the plant. But
Yeah.
also for like soil health diagnostics, like looking at the soil microbiome, trying to determine relative ratios of different microbes.
Yeah, I think it might be a little, at this current point, it might be a little qualitative, but I'm pretty sure you could get it to be quantitative.
You could probably also do quantitative by limiting dilution. Limiting dilution
Yeah.
in digital PCR, just dilute the samples out
Yep.
and measure those samples until you drop off.
So the nice thing with our collaborators, she's already incorporated, Dr. Evangelina Silva over in Biosystems Ag Engineering, has incorporated Amy's assay as a second step in her current is it Dingy or for TB? Dingy. So she was using a 1-step, okay, it looks positive, and now she's got a 2-step assay in less than 5 or 10 minutes for dengue fever, using this as kind of the second gold standard, no pun intended.
So where do we think we can go with this? I mean, let's kind of open up this discussion from Q&A to kind of just everyone chipping in and seeing where we want to go with this type of technology. Sit down.
You could use it to look at like insect vectors of diseases to see if they're carriers of citrus greening or something.
Yeah.
I would like to see it to be able to detect the pathogen before the plant actually starts showing visible symptoms.
Yeah. So that's where I think the home run is to be able to walk into a field and sample a plant and find the needle in the haystack. Then it comes into question, how much do you sample? I know with PhotosynQ, right, you guys were, after using the device for a lot, a number of measurements, you finally discovered which leaf was really the best one to sample. So that's for us is going to be, how do you sample a cucumber leaf that's that big with some kind of lancet that's the size of a pinhead? Where do you take it? Do you take multiple samples? Do you just, it's really non-invasive too. You could take a couple of punches out of a leaf, it's not for the plant.
I'd try to take a couple punches at different areas.
Yeah, just kind of go around and grind them all up and boom. You start in one corner of the field, you take your assay, you let it run for 5 minutes and you already collected 2 or 3 more and you just walk back and, okay, we're negative in this region of the field, let's go over to the next region. And 40 assays for a penny, you could spend all day, doing that for less than $1. You do an entire field for $1, exclusive at the time.
That's what undergraduate work.
Yeah, exactly. Another thing we want to do, sorry to interrupt, is just benchmark this in parallel to other assays. So the plant diagnostic clinic here, down in the head house of the greenhouse, this is USDA certified diagnostic clinic for soybean rust, for example. So when a grower detects soybean rust in their field, they send a sample to her, and they're one of 8 labs, I think, in the US that are certified to validate and say, yes, you've got soybean rust. We've got a collaboration with them, or at least it's planned, that every time they set up that PCR to detect soybean rust, and they'll wait 2.5 hours, they'll spend 10 minutes of their time and do our assay. And in 2.5 hours, they could, they're waiting on this, okay, do we have soybean rust? Well, this method says we do. Boom, yep, we do.
So, I wonder if you could do a pooled assay to probe. Let's say I'm imagining going through the field with a little vacuum cleaner. Just play with me on this one, okay? Like we're talking about taking a sample. What I'm saying, with the minimum amount, you can go through like you don't want to keep.
Yeah.
Because you're going to spread the disease, but
Exactly.
you just go along. I don't know, you're sucking up something. Yeah. And then you go, okay, you pull all that stuff down, you extract your DNA, and you say, wow, I have something here. Now you can go through and make more systematic.
You just brought up, 2 points. One is a lot of science research they're using for airborne pathogens, they'll use spore traps. So these are look like big fan blades, and they just turn at the end of the field. They have tape. So you could go pull these samples off and do the tape. You just brought up a point. Every time you guys are going through a field clamping,
Oh, yeah.
yeah, it's like sailors moving into a port. You're just running it down the field.
I actually clean my curvettes before I move between the field. I have my little Kim wipes and a little bit of.
Yeah.
We were thinking about having little sheets.
That would be
you could
handy,
pull
because
off
I
the
usually sit
Post-it
there,
note kind of things that you
because
can
I'm
do.
normally sitting there wetting my Kim wipes.
Yeah, I was thinking.
Because that's turf diseases are spread that way. They put equipment, the mowers and things, and they'll pick up the disease and they just spread it all over the golf course.
Yeah, we were worried about that. But we could take those little sheets that we were talking about, and then you give them to your thing. You could go, say what I'm saying? I don't know, how much do you pick up with taking your Kim wipe and touching it on the leaf? Do you pick up enough to assay with your?
Depends on how much is there.
Yeah.
Well, maybe you need, yeah.
Sometimes if it's not even sporulating yet, if it's not on the outside of the leaf, there's not going to be much there. If you have, I imagine you wouldn't take a PhotosynQ device and be like, oh, look, that's a fluffy leaf. I'm going to grab onto that one.
And I don't really worry about spreading
Where's the
rhizoctonia that way either, because it's very rough disease.
Yeah.
Yeah.
But other diseases, they have a lot of spores, so it just depends on the stage of the disease as far as how much would be on it. But
So for us too, the really thing we'd like to do is be able to plug into the other measurements that are being collected, right? Because plant pathogens, the environment, plant health, all of this, we can see, we've done deep, IR experiments with Dave's lab with Ruby, and we can see decreases in, photosynthetic rate in it effectively before it's symptomatic. So walks like a duck, quacks like a duck.
You could say, oh, photosynthesis is affected in a certain way, better do the
pathogen assay, right? Like go to the doctor. You've got a nasty cough there. Let's go ahead and take a culture or something.
Yeah.
Or you can take these visuals of the leaves with the camera.
Yeah.
When you start getting scraping things, then you gotta go into something.
I mean, this is a technology that really most growers, here in Michigan, what they do is they see a problem, they clip off the leaf, they put it in a baggie, they FedEx it overnight to Mary House bag, they do PCR on it, okay? That's at least 26 hours from the time he clips the leaf until she has an answer. This can be less than 10, 15 minutes. Yeah, and some of the disease, this cubensis disease, 10 days from the time a spore lands on one edge of the field, the entire 1,000 acres is gone. 10 days. So
how do you choose the region of the pathogen's genome to target? Like, if there's a commensal close relative, you might get false positive, or it could mutate and be.
Yeah, so you pick ones that are highly specific or uniquely specific to the pathogen.
How do you determine that?
Maybe like if they produce a specific toxin to their particular.
Yeah, virulence gene.
right, so well, those are highly polymorphic. Those genes are typically, those are a lot of selective pressure on those guys. But that's where we're partnering with Kevin Childs a little bit on this and doing K-mer analysis and identifying unique 25mers that are, maybe collectively, maybe it's not just one, it's 5, but the positive reaction of all 5 of those gives you confidence to say this is the pathogen. And then Amy was doing this, and she identified a primer that another lab was using to identify that pathogen, and it interacts with soybean, right? So they're like, oh, there's a problem there.
So are you looking at like non-virulent strains of other, fungi, or I guess oomycetes associated with cucumbers to validate?
That would be a goal. Right now, we're just kind of benchmarking. This has all happened in like the last 2 months, and it's just been holy crap. I feel like I'm holding on to a kind of wild horse around your house.
I'm a wild horse.
Yeah. I've got an analogy.
You can have a small tail on that.
Being drugged behind. Slow down.
Yeah, but another thing to keep in mind with this, as I mentioned earlier, is that this is mitochondrial DNA. So there's a chance for this to have more prevalence in the cell, which will allow for lower detection limits too. That might be something to look forward, going in the future, something that might be more prevalent in the cell that's easier to attack at lower levels.
So developing this bigger, what we ideally would do is like what we're doing with Carolyn Malmstrom's lab or Dave Douch's lab. Send out a postdoc or student, and we'll show you how to do it. You develop your own assay, and then go out and deploy it. I mean, that's my goal here.
So is the color change the gold interacting with the probe, or the gold interacting with any single-stranded DNA?
So it's any single-stranded DNA, but usually your DNA reanneals, and so the amount that we're seeing like that is that there's, without the target there's a lot of single-stranded DNA from the amount of probe that I'm putting in there.
Okay.
And so the ratio, it's all ratios. So it's the ratio of probe that's available, the ratio of other single-stranded DNA. And as I said, what we believe the mechanism behind this is that bubble that forms is where the rest of the nanoparticles are going when the target binds to the probe.
She did TEM, and unfortunately we didn't incorporate that data, but she did TEM, transmission electron microscopy, on gold nanoparticles interacting with DNA, both target and non-target, and it's striking. You can see the gold nanoparticles aggregating when you've got target DNA there, and so she wants to know the mechanism and understand it a little bit better. It's sorcery right now.
Well, a lot, we include the biophysics in there, but it is a lot of biophysics of what the interactions are between what we have in this assay: salt, DNA, and the gold nanoparticles. And the interaction between DNA and gold nanoparticles is still not well understood. I've read a lot over the last month trying to figure out what's going on here. Basically, every paper is well, we think it's this, but we can't really illustrate it that well. Or you have one paper that contradicts another.
Yeah, because they have DNA enzymes for detecting heavy metals, like they did mercury and copper, but you do have some free DNA if you take an environmental sample. So,
Yeah, and that was my primary concern with developing this, is like, is that around like how much single-stranded DNA is in like a crude extract.
Yeah.
But I mean, in most plant cells, you know, single-stranded DNA isn't very common.
Gets chucked in.
Yeah.
Yep.
The host, the organism really doesn't like single-stranded DNA. And with the crude lysate, you just, you know, chop it up, you're not going to shear your DNA, as well as, say, you somicated it. But the only concern that I really have with that, that's why I did the TM, is like, well, is there a secondary structure developing when your probe binds?
So would you say this is limited to like actual organism samples versus like an actual environmental sample, like a water sample or a soil sample?
No, they're doing this with water.
Okay.
Water already. Soil I haven't seen a lot. I've seen this with human samples actually, so blood, serum. They adapted it to that. So I think it's just adapting it. I think with soil, maybe it's just figuring out how this, if you do have like, let's say a small silt particle, and how would that interact with the mold. But water definitely for detecting more diseases. Sorry,
So would there be a denature step in the field, like a device that goes up to 100 degrees to allow?
I guess what the suggestion we were given was why did just a chemical denaturation. Just part of the kit in the bottom of the tube is some kind of acid, and then you immediately neutralize it after a period of time. Because, I mean, in the US, in Michigan, yeah, the easiest thing would be to plug into your cigarette lighter a small heating block in the back of your pickup truck, denature it for 5 minutes, and boom, you're done. But in, you know, third world countries, their GMC pickup trucks may not be the cigarette option.
How much of a sample do you need? 20 microliters?
For the DNA, right now I'm using 5 microliters.
So you can get that very efficiently.
Might need just like a piece of glass and
The people that gave us the suggestion are probably
Lithium battery with the solar charger on.
basically recommended, you know, basically said some kind of chemical lysis mechanism would be the easiest thing, but it's just a little drop of something that busted open the sample. That's probably maybe a little too destructive. I like heat personally.
Yeah, I mean, boiling water is something that we do everywhere.
Yeah. Maybe just build a fire on the edge of your field and put a pot of water there.
I'm talking about a tiny little sample. So
Yeah.
you
You can boil that pretty quickly. So,
Like
you know,
plastic
just
would be
while you're
just
with a
leave a
magnifying
little
glass.
water out in the sun, you should be okay. Well,
That's a marketing question right now. I mean, I guess right now what we need to figure out is what's the best way to connect to PhotosynQ or multispectrum, the device? How do we do that? What's the most logical? Is it a droplet onto the surface of a cuvette? Is it a capillary action that sucks it into a cuvette, kind of like the Beckman spectrophotometer with the zipper function, or?
I think they'll all work. I mean, the device can measure the spectrum, just like we know there, it can measure discrete wavelengths in any format you like, on a piece of paper, a leaf, a cuvette of any shape. You can put the light through a fiber if you want. As I mentioned of annealing it to a piece of glass, and you could do a total internal refraction, or all of those things are possible. I would say the simplest, because your signal's big. You got a millimeter, you're getting what, 10, 100 million, 50 million absorbance units at the peak delta. At 600, that's massive. This thing could measure down to micro absorbance.
Exactly. So, you know, yeah.
So do you see this technology as like replacing isothermal PCR point of detection devices, like the
I see it
Oppenheimer chip
replacing,
and stuff like that?
lamp assays are definitely, that's the big popular thing right now for like virus detection of bananas and citrus and things. Everybody's doing isothermal, you know, lamp assays, loop things. I think this is far superior. The cost is just, there's no amplification step in this. You're measuring native levels. You're not enriching for a positive or a negative. So that's one thing we'll work through is what is the rate of positive, you know, false positives and false negatives, things like that. But once we get a built-in standard, we'll remove the false negatives.
especially if it's not much, it's naturally low expression in the background, you still catch it.
And you put these things on some kind of, you know, mobilized chip or something like that, then you can give them cool names like here's the potato chip, the banana chip, the tomato chip, you know, and it's just all nano-based detection. Guacamole chip, the avocado. Cow chip, we could expand this to animals. Animals, why not? Chicken chips.
This goes back to this thing about heating, because this is a conversation I had with Chuck Henry. Because what you can do, now think about, so you can incorporate now chromatography steps, individual chemical reacting steps with this stuff. But this, you can actually put in also heat or light, right? Because you can turn things up, like that's, the device is a light, it may light activated things, but it can heat activated. So I'm wondering what you're talking about, the processing your sample and then really give the heat to make the single strand of DNA, and then you add this stuff, right? And I'm wondering if there's a way to automate that process using, and then monitor the absorbance changes of the spectrum changes along the way.
Like flip a little switch saying, okay, heat mode.
Well, they would do it automatically.
Yeah, yes, that's what Dave was talking about. Is there a way to functionalize the nanoparticles to photoactivate them or heat activate them?
Well, you wouldn't want to heat activate them because if they're single strand DNA that hasn't been annealed, the assay doesn't work.